मैं राजवीर सिंह शेरान, एसोसिएट प्रोफेसर इन केमिस्ट्री, SRR मोराका गवर्नमेंट PG कॉलेज झुंझुनू। फर्स्ट ऑफ ऑल, गुड मॉर्निंग टू ऑल द स्टूडेंट्स ऑफ MSc फाइनल। पिछले वीडियो लेक्चर के अंदर अपन ने एंजाइम मॉडल्स क्या हैं, कितने प्रकार के हैं, उनके बारे में डिस्कस किया था। टुडे वी विल स्टार्ट द सेल मेम्ब्रेन and transport of ion. First of all, we discuss structure and function of cell membrane. So, structure of cell membrane as shown in figure. Cells, the structural unit of living beings, are enclosed in a membrane called cell membrane. All the cellular organocells like nucleus, mitochondria, Golgi body, lysosomes, endoplasm, reticulum, ribosomes, etc. present in cellular fluid are bounded by it. In other words, cell membrane is the outer covering of all the cells and its organoids. Cell membrane measures approximately 75 angstroms in thickness. It has a three-tier structure. External and internal dense layers are separated by central low density layer. By the potassium permagnetification, it has been proved that outer layer of the membrane is polysaccharide, whereas the inner layer is protein. Central areas are made up of bimolecular layers of phospholipids. In systematic diagram, black dots represent polar layer consisting of phosphorus, carbon, nitrogen, phospholipids complex. Rods represent long chain of hydrocarbon fatty acids of hydrophobic nature. Phospholipids complex part is a hydrophilic. Long chain molecules are perpendicular to membrane. One phospholipids fatty acid layer is attached to outer polysaccharide layer and the other phospholipids fatty acids layer is attached to a monolayer of protein. Hydrophilic heads and hydrophobic tails of the central area are breathed through glycerol moiety. In animal cell membrane, glycerophospholipids and sphenophospholipids comprise polar lipids, while principal non-polar membrane lipids are neutral sterols. However, human erythrocytes membrane's outer layer is richer in cholesterol than inner layer. Cholesterol controls the fluidity of fatty acid chains of phospholipids and enhances the stability of phospholipid bilayer. So, functions of cell membrane. Cell membrane act as a barrier between intracellular as well as extracellular fluids. Through it, transport of various substances takes place either by diffusion, osmosis, or active transport. Besides, it also performs a number of other functions. An account of functions of cell membrane is given below. Suppose diffusion, molecules and ions as well as body fluids are in moving state. Motion of these particles produces heat. Greater the motion, higher is the temperature. Motion never increases except its absolute zero. This continuous movement of molecules in liquid and gases is called diffusion. Then it can be simple diffusion. Simple diffusion can occur through cell membrane by two routes: through the interstices of lipid bilayer, and secondly through water channels that penetrate through some of the large transport proteins. Diffusion through lipid bilayer. Most important factor that determines how rapidly substances diffuses through lipid bilayer is liquid solubility of the substances. Then diffusion through protein channels. Water and other lipids insoluble substances move across the cell membrane in this manner. Water diffuses through cell membrane at extremely fast rate, which is about 100 times as great as volume of red cells itself. Rate of diffusion decreases with increase in size of molecule. For example, urea molecule is 20% greater in diameter than that of water molecule. Its rate of diffusion through cell membrane is 1000 times less. So, again, facilitated diffusion. This is also known as carrier mediated diffusion, as diffusion takes place in presence of carrier proteins. The rate of facilitated diffusion also depends upon the concentration, but it reaches the limiting value of Vmax at certain fixed concentration occurring. Of the limiting rate of facilitated diffusion can be understood in terms of its mechanism. Carrier protein of protein channels has a receptor binding points. The molecule to be transported binds itself to this site, and within a friction of second, conformational changes occur, and now channel opens in the opposite direction, realizing the molecules. Because binding force of receptor is weak, the thermal motion of attacked molecule causes its break away. Then active transport. Concentration of the substance including ions is kept constant in extracellular and intracellular fluids. For instance, concentration of sodium and potassium ions. This is made possible by active transport of the ions in presence of some energy sources like ATP. Some substances which are actively transported through cell membrane are sodium ions, potassium ions, calcium, iron and hydrogen ions, chloride, iodide, urate ions and several different sugars as well as most of the amino acids. Diffusion versus active transport. Though both of these transport phenomena, former is passive transport whereas latter is active. Former is a random phenomena and makes use of normal kinetic energy of matter. It is either added or unadded by carrier proteins. But active transport involves movement of substance across the membrane through binding with carrier proteins, which helps to overcome energy gradient causes by concentration difference by at two sides of membrane and causes movement to lower concentration from higher concentration. This requires extra energy besides normal kinetic energy of the matter from additional source like molecules of ATP. Now, effect of concentration on net diffusion through a membrane. If concentration of substance is higher outside of the membrane, the rate of diffusion inverts the proportion to the concentration of substance molecule outside, because this concentration determines how many molecules strikes like the outside of the membrane each second. Contrarily, the rate at which molecules diffuses outside depends upon the concentration of molecules of substance inside. Net, the rate of diffusion is dependent on the concentration of the molecules and outside as well as inside. Net diffusion is equal to alpha C0 minus Ct, and here C0 is the concentration of molecule outside and Ci is the concentration of the molecule inside, Co minus Ci. Osmosis or net diffusion of water diffuses both sides as well as inside of cell membrane. The total number of water diffusing outside as well as inside of cell membrane is 100 times of the volume of cell itself. The diffusion of water through cell membrane, that is osmosis in both the directions, balances itself in such a way that the net movement of water is approximately zero and the volume of cell remains constant. But sometimes the concentration difference for the water occurs and movement of water accordingly occurs either out of the cell membrane, exosmosis, or inside the cell membrane, endosmosis. As a result, the former cell shrinks and later causes swelling of the cells. It's like this. So this is PEP, means in group translocation PEP plus enzyme first. In presence of magnesium, it gives rise to enzyme first P plus pyruvate. Enzyme first transfers phosphato group to heat stable protein HPR. Enzyme first PR plus HPR give rise to HPRP plus enzyme 1. HPR is smaller histidine containing proteins. Enzyme first as well as HPR are generally phosphate carrier proteins and do not show specificity. As they do not bind sugar, hence are not carrier proteins in true sense. Enzyme second is an enzyme complex containing enzymes second A and second B. Magnesium 2 plus ions are phosphatidyl glycerol. Enzyme second A is sugar specific. It carries only specific type of sugar. Enzyme second is located in cell membrane, probably in the outer region. It catalyzes the transfer of phosphorylate group from HPR to sugar. So HPR P plus sugar in enzyme second, it gives rise to sugar 6 phosphate plus HPR. Now hormone receptor interaction. Cell membrane contains receptor which recognize hormones and convey the information inside the cell to control metabolism of cell. Receptor of a number of hormones like endocrine ticotropic hormones ACTH, follicle stimulated hormones FSH, luteinizing hormones LH, prostaglandin PGE, etc. are known. Hormones receptors are generally present on the outside of the cell membrane. However, receptor for steroidal hormones are present in cytoplasm. Receptors form hormones receptor complex with hormones which travel to nucleus to influence expression of genes. First function of membrane receptor is recognition and binding with hormones. A specific receptor binds with hormone. A specific binds to a specific enzyme. The specificity of hormones and receptors is due to their complementary natures like lock and key. The hormone receptor complex thus formed results in the activation of membrane bound enzymes. Enzyme is believed to be present on the inner surface of the membrane and containing binding sites for ATP and Mg2 plus ions. Activation of enzymes, for example, aldehydes, adenylate silase, results in the generation of cyclic AMP, adenosine monophosphate from ATP. Cyclic AMP activates protein kinase, which phosphorylates a variety of metabolic enzymes, which either decreases or increases the activity of enzymes and control the cellular metabolism. Receptor may be coupled to enzyme by an intermediate coupler like phospholipids. Now, cell recognition. Leukoids, for example, in mammal, recognizes foreign cells like bacteria and kill them by process known as phagocytosis, but not the other cells. Sites of the recognition of the cells are present in plasma membrane. Probably amino sugar salicylic acid helps in the recognition of cells. Secretion. Polypeptide chains released from ribosomes are transported to Golgi body where they form secretory vessels. Secretory vessels fuse with plasma membrane through which secretion of products takes place. Antigen specificity. Glycoproteins of the surface of the cell membrane decides the specificities of cells. For instance, blood group diffraction is based upon the antigenic characteristics of cell membrane of leukocytes. Recognition of the foreign material, for example, transplanted organs too depends upon the specific antigens present on the cell membrane and is responsible for the acceptance of rejection of organs. If recognition sites of cell membrane of the organs are modified, there is acceptor of organ, otherwise body reject it. One another substance which determines antigen specificity is glycolipids. Endocytosis. Endocytosis is the process by which transportation of food and other materials takes place inside the cell by the formation of vessels on the cell membrane. It is of two types, phagocytosis and pinocytosis. Phagocytosis is the process of cutting by the cells plasma membrane of the cell fold around the material to be infused and formation of intracellular vessels phagosomes takes place. जैसे दिखाया इसमें, it is shown in the diagram. Cell giving rise to video of plasma membrane material to be nuclearized, and then it gives to phagosomes. Phagosomes fused lysosomes and subsequently get digested by the enzyme. Now pinocytosis. Intake of liquid material by cells occurs through the formation of vessels known as pinosomes, and this process is known as pinocytosis. Fluids to be engulfed may contain organic molecules as well as nutrients in the solutions. It has been shown that amoeba drinks ingests the protein solution by this mode. Proteins act as stimulate to pinocytosis. It is important to note that the protein solutions are known to be ingested by pinocytosis, because if amoeba is kept in carbohydrate solution, no pinocytosis is there, but several lines and amino acids causes pinocytosis. Pinocytosis provides cells with additional interior surface where active and passive transport can be carried out. So, thank you.